Anyway, if I'd like to introduce next to the Inquisition, Amy Sinor, Senior Research Scientist at Thermo Fisher, my old employer, who's also taken a look at the expansion of human-induced pluripotent embryonic stem cells without compromising pluripotency. I believe a big issue to the future. She also worked at Curis. I didn't know you there either. But anyway, welcome.
And I'll just be talking about some of the solutions that we've come up with for optimizing the expansion of human-induced stem cells, as well as ES cells. So as you know, stem cells have the ability to self-renew, and this self-renewal is able to go on, as well as the ability to differentiate into any cell type. And it's this unique ability that a stem cell has that provides a powerful tool for either cell replacement or to understand different disease states if you want to study basic biology. So now typically a disease usually starts with the injury or the death of a specific population of cells. So for example, in diabetes, it's the beta cells, or for different neurodegenerative diseases, it's a susceptible neuron in a specific location of the central nervous system. So for example, in ALS, as we've heard a lot about, it's the motor neurons that reside in the upper and the lower motor cortex. For Parkinson's disease, it's dopaminergic neurons. And for Huntington's disease, it's medium spiny neurons. So the best way to model these diseases is to be able to study the susceptible cell. So one approach is to differentiate ES cells into those specific cell types that are injured. So when I was at Curis and when I was at the Harvard Stem Cell Institute, what we wanted to do was undertake tasks where we would use ES cells and model different diseases by allowing those ES cells to then differentiate into the cell type that was affected in those diseases. Now these types of studies, these small molecule studies, we usually screened thousands of compounds, so you would need billions of cells. And so for example, if you were looking at a motor neuron disease, a typical cell culture assay is usually isolating motor neurons from a rat spinal cord. Well, the rat spinal cord only generates about 3,000 motor neurons. And if you wanted to study beta cells or for diabetes, it only makes up about 2% of the population. So these types of standard culturing methods are impractical if you wanted to undertake any of these small molecule screens. So biotech or pharma, what they typically do is use convenient cell types of choice. And their cell types of choice are either, if they were studying neurodegenerative diseases, is a neuronal cell line or a non-neuronal cell line. So for example, the disease that I studied in particular was SMA. For spinal muscular atrophy, it's a motor neuron disease that affects children. And the initial clinical trials were used studying compounds that worked in fibroblasts. And as you know, fibroblasts are very different than a motor neuron. So with the advent of the iPS reprogramming technology, what this allowed us was to model the actual cell types that are affected in the disease. And this is just a list of different diseases that have been modeled using iPS cells, and this is using their source cells. So most of these were generated using fibroblasts. So as I said, when I was in drug discovery, we wanted to model 2 motor neuron diseases. And so what we did was we took patient fibroblasts, reprogrammed them to generate human iPS cells, and then differentiated them into motor neurons, and looking for small molecule compounds that could alleviate some of the phenotype. And we thought that this was a better approach to understanding and ways to find therapeutics for these diseases. So some of the challenges that I had when I was in biotech and at Harvard was that we were able to generate these iPS cells, but some of our problems were that we would get very poor recovery or viability after we've frozen down these iPS cells. Sometimes we were able to get good recovery, but the next problem was able to get good expansion. So as I said, we needed billions of cells to do these small molecule screens. And so we would have issues of picking the right media and the right matrix in order to generate these large numbers of cells. And in the process, we needed to make sure that we limited our spontaneous differentiation, as well as maintaining the ability to differentiate down the 3 germ layers. And my hope was to reduce the handling time. So I spent many hours in the lab feeding the cells, spending weekends feeding the cells. And so what we wanted to do was reduce that time so we could actually do the small molecule screens and look for the therapeutics. And then we needed to also make sure that when we started the process, that the same quality and quantity of the cells that we started the process was the same that when we ended our screening. So when I was recruited to Thermo, we wanted to try to create solutions for many of the roadblocks that I faced when I was doing my workflow and my drug discovery process. And so for cryopreservation, some of the challenges that many of us faced was that we would get poor recovery from the freeze thaw. So we wouldn't get a lot of viable cells back, or the recovery was very low, so we'd have to pass the cells many weeks before we would get the numbers that we needed. And we needed to also make sure that we maintained the stemness and that our reproducibility was high. So a scientist in our group, Crystal Zellafro, created a solution, and her solution was high cryo stem. So this solution is a cryopreservation media that allows you to have high viability and improved cell recovery. It's a serum-free formulation, it's chemically defined, and it's a 2x formulation, so you dilute it 1-to-1 with your spent media. And this allows you to retain a lot of the factors and the goodies in the media that the cells have given off. And she validated this on multiple platforms. So she tested it on human ES cells, iPS cells in both formats. Typically, people grow their ES and iPS cells either on a feeder layer using MEFs or in a feeder-free format. And she also tested using cortical stem cells using mouse, rat, and human. And she, again, looked at this in the adherent as well as in the neurosphere format. So this was her protocol. She would grow either her neural stem cells or her embryonic stem cells in culture, and then she would freeze them down, again, using that 2x formulation with spent media. She would transfer them to the liquid nitrogen with -80 or the controlled rate freezer, and then she would thaw the cells and then look at them either in the neurosphere adherent format or for the ES cells, either the feeder or free formats. And she made sure that her cells did express the stem cell markers, as well as the ability for those cells to differentiate down the 3 germ layers, as well as the neural lineages for the neural stem cells. So due to time, I'll only show the neurosphere format and the adherent format for the neural stem cells. But she saw great recovery using human ES and iPS cells. So this is looking at mouse cortical stem cells. Your blue bar is our high cryo stem media, and our red bar is our 10% DMSO. And this is the standard control that people typically freeze down their mouse cortical stem cells. And this is looking at percent viability upon thaw. So as you see, she gets about 2-fold more viable cells with the high cryo stem than with the 10% DMSO. And she sees similar results whether it's in a neurosphere format or an adherent format when she used PDL laminin. And when she looks at recovery upon thaw, she froze down about 1.2 million cells, and with high cryo stem, she gets about 1.1 million cells back and about 400,000 cells with the 10% DMSO. So she's seen much better viability and much better recovery upon thaw with this media than the standard media. Now she gets similar results with 20% FBS, but the benefit of the high cryo stem media is that it's serum free. So you're not adding any endogenous factors and undefined factors that are known to be in FBS. She then went on to make sure that her cells still maintained all their stem cell qualities. So she froze down her cells, she thawed them, allowed them to grow in an adherent format, and then stained it with one potential marker, nestin, which is a neural progenitor marker. And she sees here in green that the majority of her cells within the culture are nestin positive. She then wanted to make sure that her neural stem cells could differentiate along the 3 lineages of a neural stem cell. And so she saw here, again, all of the markers are in green, and blue is for Hurst. And she saw that her cells were indeed neurons using beta-3 tubulin, that they could differentiate into astrocytes, shown here with GFAP, as well as oligodendrocytes, shown here with O4. So Thermo Fisher Scientific has a solution for your cryopreservation needs. We have a high cryo stem. This formulation, as I said, is serum-free, and it has much better viability and cell recovery than 10% DMSO. And it can be used with both neural stem cells as well as human embryonic and iPS cells. So the next issue in my roadblock, as I said, was the expansion. And so we wanted to find a solution for this problem. And so in this process, these were some of the things that we were looking at. And we were looking at, ways to promote expansion. So could we get better expansion than some of the commercial medias that are existing out there? And at that time, we wanted to make sure that if we were able to promote expansion, that those stem cells still maintained their stemness. So they still had the right morphology, they had tight borders, high nucleus to cytoplasmic ratio, they had limited spontaneous differentiation, as well as have the capacity to differentiate along the 3 germ layers. And our hope, again, was to limit the amount of time that you're spending just setting up the cultures and feeding them every day. And we wanted something that was easy to use with minimal lot-to-lot variability as well as adaptation to this media. So we have 2 solutions. We have one for the camp out there that is still using feeders as well as a feeder-free system. And for right now, I'll talk about the feeder system. So we validated this media internally using 2 ES lines and 1 iPS line. We grew them using mouse embryonic fibroblasts, so that was our feeder layer. We fed the cells daily and every other day. And then we passaged the cells weekly, and we looked at 5 to 10 passages, depending on the cell types. And we used HiQ-TASTE to dissociate the cells down to single cells, as well as used manual dissociation. So that way we kept the cells either as single cells or as clumps. And then this is just our experimental parameters that we looked at. We looked at morphology daily, we looked at recovery from thaw, we looked at expansion, so we were looking at SOX and OCT2 positive cells at every split, and we made sure that the cells still had the ability to differentiate down the different germ layers and that the karyotype was preserved. So what we saw when we thawed the cells directly into high cell stem and into the standard ES growth media was that our cells recovered about 2.8-fold more OCT4 positive cells than the standard media. So this allows you to expand your cells much faster because you've preserved many more of the stem cells from the outset. And if you looked at the morphology of the high cell stem cells versus the standard ES growth media, you don't see much difference. We also stained the colonies for OCT4. So here's our high cell stem colony stained with OCT4. And as you see, all of the cells within the colony are OCT4 positive. And this is very similar to what you see with standard ES growth media. What we did see was that depending on certain cell lines, we did see a flatter morphology than what we did see with the standard ES growth media. But these cells still maintained all the normal properties of a stem cell. We wanted to make sure that our media was not dependent on ROCK-I. So this might seem like a complicated slide, but this is looking at the number of OCT4 positive cells, and then this is looking at the fold increase of OCT4 positive cells normalized to the standard control without growth ROCK-I. So these are cells that are thawed into our media or into the standard ES growth media, and you see about 2.5-fold more OCT4 positive cells compared to the standard ES growth media. And what's interesting is that you see almost the same number of OCT4 cells when the standard media has the ROCK-I. And as you know, ROCK-I is known to promote survival. So this suggests, and we still have to do further studies, understanding the mechanism on how this is working, but it's perhaps that our media is promoting better survival than the standard ES growth media. If we looked at the theoretical expansion curve, so this is if you took every single cell within your well and expanded it over 10 passages, you would get, and this is log scale, about 8 orders of magnitude more cells in the high cell stem if it's fed daily or every other day in comparison to the standard ES growth media. So for example, if you were setting up the kinds of studies that I was setting up and you needed 1 billion cells, in our media, it would take you about 3 weeks to generate 1 billion cells. While in the standard media, it would take you about 5 weeks. So this allows you to set up your experiments much faster and allows you to save time where you're just not feeding and expanding your population. We then wanted to make sure that throughout the 10 passages that the cells expressed OCT4 positive cells, and we also looked at the percent OCT4 positive. So this is looking at the total cell count, and this incorporates the MEF population, because again, this is on feeders, the differentiated cells, as well as the stem cells. And if you look at the cells that are fed daily or every other day in the high cell stem media, compared to the standard shown here in red, you see many more total cells or OCT4 positive cells in comparison to the standard media. And this holds true for over the 10 passages. And what's interesting is the every other day and the daily fed cells are equivalent. So this allows you to take the weekend off. So typically what we would do is we would feed the cells on Friday, and we wouldn't touch them until Monday morning, which was great. What's also interesting is this first set of bars is the cells that are coming directly from the thaw. And as I said, they do much better from the thaw in our media than in the standard ES growth media. And when you looked at the percent positive cells, you also saw a greater number, indicating that there was less spontaneous differentiation in our media than in the standard media. When we looked at its ability to maintain differentiation, we saw that there was no difference between the cells grown in our media for 10 passages versus the standard ES growth media. So they were able to differentiate along ectoderm, endoderm, and mesoderm lineages. When we looked at karyotype at passage 10, we saw no karyotypic instability. So this made us very happy. We then went on to look at creating a formulation for the feeder-free system, as we know that researchers are now moving more towards a feeder-free option. So again, we looked at the same ES and IPS lines. Now we were growing them on vitronectin. We passaged the cells at shorter intervals every 4 to 5 days because they tended to grow much faster in the feeder-free system. And we used EDTA to differentiate the cells down to single cells. And again, we studied all of the same hallmark features. So with the feeder-free media, we saw much better recovery from thaw in our media than in the standard media. So we saw 3.5-fold more OCT4 positive cells in our media than in the standard media. So again, this allows you to recover more cells initially so that your expansion can happen much faster. And again, the cells look very normal in comparison to the standard ES growth media. When we stained our colony for OCT4, we saw that our colonies, that all of the cells within the colony were OCT4 positive. We saw very tight borders, we saw minimal spontaneous differentiation, and they looked very similar to the standard ES growth media. We again looked at that expansion rate over the 10 passages, and we saw a 6 orders of magnitude more cells in our high cell stem media than we did in the standard ES growth media. So again, if you needed to generate those billion cells, you can get it in about 3.5 weeks versus the 5 weeks. So again, it's saving you time, it's saving you money as well. We also looked at the total cell count and the OCT4 positive cells over 10 passages, and again, we saw much better results in the high cell stem than in the standard media over the 10 passages, and we saw many more OCT4 positive cells. And we saw very minimal spontaneous differentiation. So we were almost getting 95 to 98% of the cells were OCT4 positive. And we looked at this data also using SOX2 as a marker. Again, we looked at the ability of those cells grown in the high cell stem FF media to differentiate, and we saw no problems with differentiation. So the cells could differentiate into beta-3 tubulin cells, FOXA2, and SMA, and that's similar to what we saw with the standard ES growth media. We also wanted to make sure that the cells could maintain their karyotype for 10 passages, and again, we saw no karyotypic instability. So this protocol is very easy to use. You don't have to change anything that you've done standard, so it's very standard. You can either play it on MEFs Or in the Feederfree, we validated it using vitronectin. The only difference that you probably have to do is plate much fewer cells. So our split ratios were sometimes 1 to 50, 1 to 100, because they would grow so much faster than in the cells that were growing in the standard media. And I think we were also maintaining a much better survival at each split. And so that's why the ratios were much higher than what we typically would have seen in normal cultures. For the feeder dependent, you can feed the cells daily or every other day. For the feeder independent, you have to feed it daily. And again, as I said, we dissociated the cells down to single cells, or we did manual dissociation. So it's a very standard protocol. So I hope I've shown you that we can meet many of the challenges that you might face when you're culturing your stem cells. So we have much better recovery from the freeze thaw. It's about 3-fold more than the standard ES growth media. You can generate more cells in fewer passages while maintaining their stemness, so we have limited spontaneous differentiation. We still preserve the ability to differentiate down the 3 germ members and maintain normal karyotype. And we can reduce the handling time. So this media is very simple to use. It comes in 2 components. You have your basal media and your supplement, and all you have to do is basically dump your supplement into the basal bottle, and then you have a complete media that's stable for 4 weeks at +4. And so here are some of the solutions that Thermo has for culturing stem cells. I didn't go into detail about the high cryo, which is an AOF formulation that you can use with general cell types. It's also a 2X formulation, but if you'd like more information, please come visit us at the booth. We have a high cryo stem, which I explained was our serum-free media, and then we have our 2 versions for our expansion and growth of embryonic stem cells.